Welcome back to part 3 of modeling and estimating genetic and environmental components. Now that we've established that we can estimate heritability with data from twins, we focus here on modeling genetic and environmental sources of variation in a single trait. Here is a brief outline. First, we'll talk a little bit in detail about what we mean with the ACE and the ADE model. We explain the difference between the path coefficients and the direct variance estimation approach, and then show the key commands of the univariate twin model script for a single trait, as well as an alternative specification. We talk a little bit about how to interpret the goodness of fit statistics as well as the parameter estimates, before highlighting some additional issues, including how to analyze data that are either binary or ordinal, how to include covariates, how to test for heterogeneity with a focus on sex differences, and finally, how to include information from other relatives such as siblings and parents. So let's start with the ACE or ADE model. So the focus here is on what are the contributions of these potential sources of variance to the total variance of the trait. Note that we might interchangeably use E and VE11 in these slides to simplify some of the notations. You might remember from our previous presentation how we get to estimate the variance components. And we've established that the MZ and the DC correlation either can be equal to one another, when that would imply that the shared environment is primarily responsible for those correlations. We've established that we can assume that genetic influences are important when the MZ correlation is greater than the DC correlation. But if the DC correlation is either greater than half the MZ correlation, or half the MZ correlation is less than the DC correlation, that would imply or suggest shared environmental influences in addition to additive genetic factors. However, in the reverse situation, when half the MZ correlation is greater than the DC correlation, or in other words, the DC correlation is less than half the MZ correlation, that would suggest additive and dominance genetic factors contributing to the phenotype. But clearly, the DC correlation cannot be at the same time both less than or greater than the MZ correlation. So that leads us to the fact that models with all 4 sources of variance aren't identified, as the classical twin design only includes 3 unique statistics: the total variance, as well as the MZ and the DC covariance. So it allows us to only estimate 3 parameters simultaneously. And so typically that would be A, C, and E, or A, D, and E, as C and D are confounded, given they have opposite expectations. So let's then see how we would draw an ACE model. We have the phenotypes of twin 1 and twin 2. Now we are assuming that these phenotypes are accounted for by 3 sources of variance, A, C, and E, that we directly estimate here. And we know that MZ twins share all their additive genetic variance as well as their common environmental variance. So the expectation for the covariance between twin 1 and twin 2 is A plus C, or VA plus VC. Whilst for the DC twins, the expectation of the covariance, which you can work out with the rules of path analysis, is a half A plus C. Note that the expectation for the variance is the same for both twin 1 and twin 2, as well as MCs and DCs, and is A plus C plus E. The alternative model that we can fit to twin data is the ADE model. And again, we can simply use the rules of path analysis to work out the expectations. And in this case, for the MZ twins, the expectation for the variance is A plus D plus E, where that of the covariance is all of the additive as well as all of the dominance variance, so A plus D. We can work out the expectations for the DC covariance from the biometrical model, and it turns out that DC twins on average share half their additive genetic variance and a quarter of their dominance variance. This leads us also to talk a little bit more about the difference between the specifications of the model in terms of path coefficients and direct variance estimation. So if you've read papers from a couple of decades ago, most of those would have used the path coefficients approach, which bounded the variance components to be positive, because we don't really expect a variance component to go negative. However, it may make incorrect inferences when comparing alternative models. So instead, we now recommend that one uses the direct symmetric variance estimation, which allows the variance components to be negative, but then as a result makes more correct inferences when comparing alternative models. When you have a model that resulted in a negative variance components, it sort of indicates that that may not be a good representation of your data, and so it suggests that you should fit the alternative model. However, the good news is that you can actually just calculate the dominance component, even if you fit an ACE model, and alternatively, you can estimate or calculate the shared environmental component if you had fitted an ADE model. And I give you here the derivation of these variance components in the two different scenarios. Let's move on and focus on the key statements in the univariate twin model script that allows us to estimate either the ADE or the ACE model. We'll start with the ADE model. Here, this is the same path diagram, except that I've separated the MZ diagram and the DC diagram, as they are really fitted separately to the MZ and the DC data. So this is a multi-group script. First of all, we specify the means, and we've established that we can equate them across twin order and zygosity. So we use the same label for each of the means, and we specify this using an MX matrix command. And this is a 1x2 matrix. Next, we want to specify a matrix for the additive genetic component, as well as for the dominance and the unique environmental component. And all of these are 1x1 matrices, where we freely are going to estimate these three components. We can then use an MX algebra statement to generate the expected variance as the sum of the three variance component. So we do this with an algebra statement below. Similarly, for the MZ and the DC covariance, we can specify those using MX algebra as the sum of either VA plus VD for the MZ covariance, or a half VA and a quarter VD for the dominance variance. Now, our expected covariance matrix for either the MZs or the DCs is a 2x2 matrix, which we generate by using the cbind and the rbind commands in R to put together these 1 by 1 matrices into a 2 by 2 expected covariance matrix for MZs and DZs. Now that we've worked out how to do this for the ADE model, of course, it's very similar to do this for an ACE model. Here's the diagram, the means, the variance components, total variance, expectations for the MZ and the DZ covariance now in terms of the ACE model, and the expected covariance matrices. So this is one way in which we can set up the model so that we can freely estimate these variance components and then test whether or not they contribute significantly to the variance of the trait of interest. However, there is also an alternative way in which we can specify this model that might be more comparable to what you're familiar with from the Haseman and Elston regression, which regresses the phenotypic correlation on the genetic relatedness. So let's recap. This was the way in which we set up the model and generated our 2 by 2 matrices, which look like this, where I've now used an alpha here to indicate a 1 for MZs and a half for DZs to distinguish between their expected correlations for their genetic component. So if we rewrite this model and look specifically at these variance covariance matrices, but focus separately on the A component, the C component, and the E component, then we see a certain structure in these matrices. The A component can basically be rewritten as our VA additive genetic variance matrix times a matrix of the genetic relatedness. Where we know that the genetic relatedness for MZs is 1 and for DZs is 0.5. If we look at the C matrix, you see that C contributes not only to the variance but also the covariance. So rather than that's specifying this as a symmetric matrix, we can specify this as a full matrix, where we multiply the variance component for C with the shared environmental expectation of the correlation. Finally, the E component only contributes to the variances and not to the covariances, so this can be specified as a diagonal matrix. So if we generate all these new matrices with the specific type, symmetric for the genetic ones, full or unit for the shared environmental one, and an identity matrix for the unique environmental correlations, then this gives us the same expected covariance matrix for the MZs and the DZs, but now expressed in a way that is similar to how the Haseman-Elston expectation is generated. Of course, given that we have both MZ and DZ twins, in addition to estimating the heritability, we can also estimate the shared environmental variance component in addition to the residual unique environmental variance component. Of course, we can go a step further if we think about the fact that instead of using a matrix with a specific value of 1 for the MZs and a half for the DZ covariance between the genetic components, we could put those values into a definition variable. In this case, we call that rel, where we give it a fixed value of 1 for MZs and a half for DZs, and then we can reduce the matrices to just one RA matrix that now has specific values for the MZs and the DZs. Going a step further, instead of actually using the expected relatedness for MZs and DZs, if we have genotypic data for these twins as well, we can use the actual observed relatedness, especially for the DZs, of course, where that varies around 0.5, and get an estimate that becomes much more similar to what we would be doing if we were to estimate SNP heritability. Again, of course, with the addition that we also estimate the shared environmental component. So hopefully here I've shown that our classic twin model, especially when we have genotypic data as well, allows us to fit a model that is very similar to how we estimate the SNP heritability. So now that we have established how to specify this model and how to fit it, let's see what output we get from running this model in OpenMX. So typically, we start by comparing our genetic model, either the ADE or the ACE model, to our saturated model. That one had 10 parameters. Our genetic model only has 4, including the 3 variance components and the mean. And you see here that when we take the difference between the likelihoods of these two models, we get a value of 7.5 for 6 degrees of freedom, with an associated p-value of 0.27, which is clearly indicating that this ADE model does not fit the data significantly worse than our saturated model, and so we have an acceptable model fit. We can go a step further and test whether each of these variance components are indeed significantly different from 0. We do that by dropping these components one by one from the model, except that we would always keep the unique environmental component into the model, because obviously that also includes residual measurement error. Further, when reducing the ADE model, we would typically not fit a model with just the dominance component, as it is not biologically plausible. However, we can test the significance of this dominance component by dropping it from the model. And when we do that in model 2 here, you see that we went from a minus twice log likelihood of 4063 to 4067, or 4.21 for 1 degree of freedom, which gives us a p-value of 0.04, just below the significance level of 0.05, which would indicate that in this case, for this particular example, the dominance component is indeed significant and we cannot drop it from the model. So let's look at how this translates into the estimates of the parameters. So here we have provided them for all 3 models, even though we've already established that the ADE model is the best fitting model, which we could have also concluded from looking at the AIC, where the model with the lowest AIC is typically considered the most parsimonious model. And so you see here that we have on one hand included the unstandardized variance components with their confidence intervals. And on the right-hand side, we have divided each of the variance components by the total variance to give us the standardized components. And you can see that even when we ask to estimate confidence intervals, we see that both or all 3 confidence intervals do not include 0, indicating that each of these variance components indeed contributes significantly to BMI in Australian females in this sample. The proportion of variance accounted for by the additive component is 0.41. The one for the dominance component is 0.37. So combined, this gives us the broad heritability, which is estimated at 78%, where the narrow heritability, or the additive genetic variance over the total variance, is estimated at 41%. We will go through examples of this in our practical during the workshop. Now, we have so far analyzed a continuous trait, but how do we deal with binary or ordinal traits? So often, our measuring instrument can only discriminate between two or few ordered categories, such as the absence or the presence of disease, and so we end up with counts, or the number of individuals in these categories. Typically, we assume that the observed binary or ordinal variable is indicative of an underlying, latent, and unobserved, continuous, normally distributed variable called the liability. And this is the concept of the liability threshold model that was introduced earlier as well. So here we show a visual of this underlying normal distribution where we have arbitrarily put in a threshold that distinguishes those above the threshold, say those with a disease, from those below the threshold. So it turns out that these cell counts of these two categorical traits can be measured in monozygotic and dizygotic twins, can be represented in contingency tables. And these can then be translated into proportions, which is depicted here in this table, as well as in this bivariate distribution, which has been chopped up in the 2 dimensions to indicate those that are concordant, affected, concordant, unaffected, or discordant. And it is these observed proportions that relate to those expected under the bivariate normal distribution with a particular correlation, as well as specific thresholds, that allows us to then estimate the A, C, and E, or AD and E components from the MZ and the DZ tetrachoric or polychoric correlations. Now, often we know that there might be important covariates that should be taken into account, and the ones that we typically think of are age and sex, and we're going to focus primarily on sex here as an example, but there are potential other covariates that might have to be included. So, of course, we would normally do this with a simple regression that allows us to capture these differences in mean levels of a trait, say by sex. But in addition to mean levels, there could also be differences in the variances across sex, or both in mean and variance. And if we ignore these mean differences, they can even induce variance differences. So it is important to include these covariates for sex or for other potential important covariates into these models. And it is really analogous to including the constituent terms in an interaction model. So typically we do this like we've seen before with the SNP in the linear regression model as definition variables, and then adjust our expectation for the mean accordingly by adding in these extra terms for the regression on sex and age. So we've talked about mean differences, but also potentially variance differences. So if there are variance differences, say between males and females, That means that these variance differences could be due to differences in the additive genetic variance or into the shared or unshared environmental variance components. And so, in addition to sex, there could be other independent variables that actually influence the trait to different extent in different groups. Males, females, urban and rural, or twins raised in urban and rural environments. And when we try to establish whether there are indeed differences between the variance components in different groups, we talk about quantitative sex differences. So we could, for example, test whether the contribution of genetic factors is greater or smaller in males or females. To do this, of course, we would need to have data on both males and females, and instead of a 2-group analysis, we now would do this in a 4 or 5-group analysis, where we estimate separate A, C, and E components in males and females. You notice that there is an additional group in this case. When we have sex, we also have a group of opposite sex twins that allows us to answer further questions about potential sex differences that go beyond whether or not these sources of variance explain a different amount of variance in males and females, but maybe there are different kinds or different nature of independent variables that influence the trait in different groups. And this would be referred to as qualitative sex differences. But it requires pairs that are discordant for group membership. So in the case of sex, we have opposite sex twin pairs, but of course, they are always DZ twins. And so we can specify not only different variance components in males and females, but also estimate whether the same or different genes contribute to the trait in males and females by adjusting the expected covariance in opposite sex twin pairs. Finally, these models are not limited to data from twins, MC and DC twins, and we could add other relatives that provide additional information to estimate the sources of variation. When we include siblings, it could be either as separate groups, or we could have twins who have an extra sibling, and then we just increase the size of the expected covariance matrices with the extra SIP. Of course, we can then again use either the expected relatedness for full siblings, which is 0.5, similar to DZ twins, or if we have genotypic data, obviously there would be a distribution of this pi hat around 0.5 that we could also include in our analyses to get more precision of the estimate of the genetic variance or the heritability. In some instances, we also have data on the parents of twins, which then opens up a whole new range of possibilities in terms of additional sources of variance that can be estimated. Basically, what they allow is that they give us some extra statistics to say simultaneously estimate the additive and the dominance genetic component, or further break down the shared environmental component into those sources that are shared within generation or within the offspring generation, in this case, the twin generation, where we could even separate out the twin effects from the sibling effects if twins maybe share more of their environment than siblings do. But the main difference is that we can now also look at cultural transmission or environmental transmission from parents to offspring, in addition to genetic transmission. And if parents both contribute to the environment of their children through cultural transmission, and the trait is genetic, so there is genetic transmission, then this would allow us an estimate of genotype environment covariance, which is basically also termed genetic nurture. In addition, parents, of course, would provide an estimate of the spousal correlation, which then lets us estimate whether or not there is random mating for the trait of interest, or whether there is some significant or non-zero phenotypic or other types of assortment. So, as you can tell, by adding these other relatives, suddenly we can estimate more parameters, which may provide us a more realistic estimate of these different sources of variation. And this is an area that Dr. Lyndon Eaves really established. Well, to be fair, this is not the only area of where he has made an enormous contribution to the analysis of genetically informative data. If you think of any extension of these models, or even the basic model to start with, the multivariate extension, which we'll talk about next, genotype by environment interaction, and so many other topics. Most of the time, Lyndon has already come up with ways to solve these additional problems. So a huge thank you, and fond memories of working with Lyndon on these models. Thank you.